Good morning, and thank you for turning up in such great numbers today. It's really fantastic and very invigorating to see so many of you being engaged in this cause. So I see Miriam waving at me. And you can hear me? Okay. So the slides are behind me, so I'll point behind me. I'm going to talk about the microbiome and its potential role in Parkinson's disease today. And the subtitle is Getting to Know Your 100 Trillion Best Friends, because it is, if you count them all one by one, it is probably a number of about 100 trillion different microbes that live in and on us. Let's start with number one, and by lunchtime we should be by number 50, and we'll work our way through. So I have a few disclosures. First of all, that this research project, although funded by several grant agencies, one of them is the Parkinson Society BC, and I'm very grateful for that. Without your support, we could not do any of this research. So this is your research. You're actively engaged in that, not only by supporting it by voting for the motion and with the financial implications as well, but also, of course, being there, participating in studies. So I really, really thank you for your commitment there. The Pacific Parkinson's Research Institute and the Center for Brain Health at UBC are other funding sources. So what are we going to do? I'll give you a brief introduction into the microbiome, which has become quite a buzzword. We're going to talk about how the microbiome might actually influence the brain and vice versa. We're going to look at recent developments in Parkinson's disease, and then also what we're doing out at UBC. So this is a cartoon of evolution, and if you look at that, let me just see them. That must be the pointer. No? Can you see a pointer? No pointer. But I can't see the pointer. In any case, it doesn't matter. So the message here is the bacteria joined the world billions of years ago, and we humans joined much, much later, really last minute. And as we evolved and developed, the bacteria were already there, and they evolved and developed with us. So in other words, we've always been absolutely intricately related to the bacteria, and we've always been relying on them. And therefore, it is not by coincidence that the bacteria and the microbiome in general are actually providing us with a lot of positive function without which we could not live. So for example, they defend us, the good ones, defend us from the bad ones, the harmful microbes. Now we all know that if you get, let's say, diarrhea, then the bad ones have taken over. They're breaking down nutrients. In other words, they're helping us to digest our food. And not only that, they're also producing vitamins that we can't produce ourselves. So such as vitamin K or vitamin B12, which we vitally need to live. They actually produce hormones as well, and they can actually, and we will get back to that, produce other metabolites that can act as messengers in our bodies. They're also involved in processing external agents that enter our body, such as medications, but also things like pesticides or pollutants, which of course is very important. They have an influence on gut motility, so in other words, on how our gut moves, whether we've got constipation or the example of diarrhea. They train the immune system. Without a functioning microbiome, our immune system is not going to function well, it's not going to be trained, and they also can influence behaviors, and that, of course, is the topic of our talk today. So there are now a few words that I've been using several times, and I should probably explain what I mean by them. So first of all, the microbiota. Microbiota, that term refers to all the microbes living in and on us. So that is anything from bacteria to viruses. There's something called archaea, fungi. So all of those together are called the microbiota. And if you were to weigh them all, they would weigh somewhere between 1 and 1.5, maybe 2 kilograms, which is just a bit more than our brain weighs, actually. So it's rather surprising that we haven't really paid all that much attention to them in the past. Their highest density, not surprising, is in the colon. So what we found in the feces, actually a large part of that is microbiota that we're getting rid of. So how do we know who's actually there? Well, part of the problem is that a lot of those microbes need what we call anaerobic living conditions, meaning they actually do not like oxygen. They thrive in environments where there is not much oxygen, and if you expose them to a lot of oxygen, they die. They do not do well. So the problem with that is that once they leave their space, in this case, for example, the colon, so the gut, they're not going to survive, and we can't really culture them, which is the traditional method of knowing which microbes, particularly which bacteria are there. Now, fortunately, they all have their own genetic information, and we can use that genetic information to analyze the specific genes that we know are specific to particular types of microbes, and with that, we can then say, okay, based on the genetic information that we can analyze, these specific microbes are there. And it's about 10,000 different ones just for the gut. And therefore, the term microbiome comes into play. And microbiome really refers to this collected genetic information that the microbiota carry. Interestingly enough, if you were to count your different genes that we all as humans carry, and we count the vast amount of different genes that the microbes carry in us, then we are completely outnumbered, so by about 100 to 1. So there is much more genetic information, different genetic information in the microbes than our bodies actually. Fortunately, and Stefano will talk to that later as well, with the advancement of genetic analysis, so high throughput genetic sequencing, we're now actually in a position where this vast amount of information can be analyzed and processed. Now, what influences what kind of bugs I've got in my gut, in my nose, on my skin, etc.? The first point of colonization, so where after the baby or when the baby's in the womb and then is being born, and the first point of colonization actually is birth itself. And it is very different between having a C-section, which is much more of a sterile procedure, than having to go through a colonized birth canal and spending maybe a few hours there kind of trying to get out. And this has led actually to this insight now, has led to new studies that take swabs from the mom's vagina, so to get a sample of the microbiome of the microbiota living there, and to swap the baby if it has been born by C-section, so that it gets a bit of a head start with that colonization, because it is now recognized. And this isn't just all icky and yucky and dirty, but this is actually absolutely vital for us. And in order to have a healthy microbiome environment, you need that process. Then, of course, there are the host genetics, and again, we'll hear more about that, but certain personal makeups of the immune system and of your personal makeup as a human will likely influence which microbes are more likely to survive and thrive. Your health status, our lifestyle plays a role, anything from sleep and exercise, etc. Use of medications, particularly, of course, antibiotics, but other medications as well, COMT inhibitors, for example. Diet is the biggest one by far. And diet, this would be another whole talk, but diet does play a large role, and generally speaking, all I say about that, if you have a healthy diet, and so Mediterranean style diet with a lot of fresh fruit and vegetables, you're also feeding your microbiome well. So when you for the next meal, think of feeding your microbiome and keeping it happy. So I realize that most of you won't be able to read this because it is too small and you're too far away. But just to say, so those are conditions currently linked to the microbiome in one way or another. Of course, the list particularly includes gastrointestinal disorders, so gut disorders, but it also includes cardiovascular, so heart and vessel disorders. And increasingly, it includes conditions that somehow are related to the brain. Some of them, such as Alzheimer's disease, have entered that list fairly recently. Parkinson's disease is, of course, the one that we're talking about, and that is probably one of the ones where we have the most research. And then we've got autism, depression, anxiety as well. So how can microbes influence behavior? Well, one very common example that we all know is what we call sickness behavior. If you're feeling poorly because the microbes, the bad ones, have taken over, all you want to do, behavior-wise, is curl up in bed. In other words, be socially isolated. Your mood is probably, our mood is down. We are having a lack of appetite, so we're not eating, another behavior, and we're feeling really fatigued. So all of those are things that are mediated by the brain, and the microbes, through the inflammation, through the immune system, can directly influence how we're behaving because we're curling up in bed. Now there's another one, and this is an example. I just give you that as an example, which I always find absolutely fascinating. There is a microbe, a parasite called Toxoplasma, and that parasite lives in cats. That's the primary host. But it can also live for a short time in rodents, otherwise known as mice and rats and so on. And what happens is the Toxoplasma infects the cat. The cat then has the eggs of the Toxoplasma, or the Toxoplasma in its feces, and that is then being eaten by the rats and the mice. Then the Toxoplasma is in the rats and mice, and of course it now needs to get into the cat again. In order to do that, the Toxoplasma has developed the ability to make the rats or the mice fall in love with the cats. Now, that is a bit of a tricky situation, leading to this all-consuming love affair. So in the upper corner there, you see the mouse and the cat very close together, and then what happens next is what you predict to happen. The cat does what it always does, and the Toxoplasma is in its new host and happily thrives and has spread to the next animal. So in other words, it can influence the behavior of a mammal. And with that, I introduce you to what we think might be going on, what we hypothesize is going on when we talk about the gut-brain connection. So there are several pathways and steps involved here. And the first one is we think maybe there's some imbalance of the microbiome in the gut, and that imbalance leads to what looks like more inflammation. It leads to more leakiness of this brain, of that barrier that the gut usually presents, so that maybe other metabolites can come in, that the microbes themselves might produce more and make the immune system produce more pro-inflammatory factors that then influence both the entire body but also talk to the brain. The microbes will also have an influence on how medications and toxins and pesticides are being processed and how they influence us. And then the connection to the brain happens either via the immune pathways, or it happens by substances that the microbes produce. So for example, also dopamine and serotonin, although this is staying usually outside the brain. And then there is a highway called the vagus nerve. The vagus nerve is connecting the brain and the gut directly. It's like literally like a highway. It's a hardwired, it's not a Wi-Fi system, this is hardwired from the gut to the brain. It works both ways. So communication is from the gut to the brain as well as from the brain to the gut. And we'll see why that might play a role as literally a highway for communication here. So this is what we think is likely happening. But this is a hypothesis, and we need to do an enormous amount of research to fill in all the gaps in that picture, to really be able to prove that this is what's happening. We've got some pieces in the puzzle, but we are far from having the full picture, and that is why we need to do the research. So recent insights into Parkinson's disease. So why do we investigate this at all? So in the clinic, and I think this is a really important point, as clinicians, where we work with you all the time, we of course hear your stories and we hear what is going on, and we take those questions and those gaps to the lab and try to figure out some of these answers. Yeah, and so one thing that we see over and over again is that constipation, as well as a lack of sense of smell, although this might not be entirely obvious to everybody, but both of these are occurring very, very frequently in the vast majority of people. And really importantly is they occur often very early on, often years, sometimes decades, before any of the motor symptoms occur. So something happens in the gut and the nose much before anything happens with the brain. Of course, those organs are at the exact interface of where the host, that is us as humans, and the environment interact. And importantly, those surfaces are heavily populated by microbes. As we just heard, the colon, for example, has the highest density of microbes living in us, and the nose, of course, is populated as well. So they're sitting right at that possibly very, very important place there. Anxiety, depression, sometimes mild cognitive changes or fatigue are also very early symptoms, and they might also occur before any motor symptoms occur. And as we saw earlier, anxiety, depression, fatigue, think of sickness behavior, are all related to the microbiome in people who do not have Parkinson's disease. So you kind of wonder, maybe there's another connection. And then there's the vagus nerve again. So epidemiology are these huge, large population studies. In this case, it's a registry in Denmark, where many of the Scandinavian countries have these registries where health events are being captured on a population basis. So this is looking at tens of thousands of people. And in the past, there was a treatment where they cut the vagus nerve. So they blocked that highway, cut that highway between the gut, in this case, particularly the stomach and the brain, to treat ulcers in the stomach. Now, of course, we know that it's actually a bug that is causing those ulcers, but, just by the by. So there it is again, the bugs. But they cut this vagus nerve, and the people who had the vagus nerve completely cut actually were at a much lower risk of developing Parkinson's in the years to come. So there's this little graph, which you probably can't see, but there are two curves going like this. This is your normal risk of Parkinson's, and this is the reduced risk if you have the vagus nerve cut. And you have to have it cut completely, because if you only partially cut it, that doesn't work. There's still something going through. We don't know quite yet what that something is. And then if you look at tissue, you also get a few more hints of why this might be important to look at the microbiome. In Parkinson's, mild inflammation in the gut has been demonstrated. There's also been a demonstration of reduced barrier function, so the gut isn't quite as tightly connected as it should be, but things can come through more easily, and those could be toxins, but this can also be specific microbes that just kind of invade this outer layer. Also alpha-synuclein, which is this protein that tends to clump up in the brain, but and then is one of the, if not, well, it is the major component of Lewy bodies, which are the hallmark of pathology in Parkinson's. This alpha-synuclein, the alpha-synuclein deposits can be found in the gut early on, before it can actually be shown in the brain. It's not entirely specific for Parkinson's. Not everybody who's got it in the gut will have it in the brain, but it is potentially a very early event. In addition, we'll have inflammation both in the body as well as in the brain in Parkinson's that we can show, and the question is, where is that coming from? Could that, for example, be triggered by microbes? And with all of that in mind, the Parkinson's researchers decided we got to look at those microbes and see whether they might contribute to those processes. So there were several what we call cross-sectional, so at one time point, not followed up in future and done a few times, but just done at one time point in every person who was participating. There are about 5 of those studies published now, and they all showed that there were slight differences between the composition of the microbiome in people who have Parkinson's and in those who don't. The differences didn't completely match between the different studies, and there are many reasons for that, and that is one of our tasks, of course, to figure out why that would be. But the general message we're getting from those studies is that there's probably, with that particular composition of the microbiome in Parkinson's disease, it seems to drive inflammation more. There are more pro-inflammatory species present and less anti-inflammatory species. Seems to be a bit less bacterial diversity, and diversity is always a good thing, or nearly always. So the spectrum seems to be a bit narrower in Parkinson's. And there are certain components, such as, for example, short-chain fatty acids that the bacteria produce that seem to be reduced in Parkinson's, but we'll get back to those. That story isn't quite clear yet. So with that, I want to introduce you to a very elegant experience, really a series of experiments that were deducted recently and published in the last few months. And the main players are, we've got our mouse, but it's one that is genetically engineered to overexpress alpha-synuclein. So they make much more alpha-synuclein, which is this protein that then clumps up and is very vital and important for the process of developing Parkinson's symptoms. Then we've got all our microbes, which are in the middle there. We've got, in the left lower corner, we've got some neurons and some microglia. Microglia are immune cells that reside in the brain. So they are the ones who clear up, but they also cause inflammation or can contribute to inflammation. We've got the short-chain fatty acids again as one of the products that the bacteria can produce. Yeah, and then we've got alpha-synuclein. There's a Lewy body there in one of the cells, in the dark blot there. So what they did, they have these mice that are overexpressing, so making much more alpha-synuclein than they should. And these mice are, we know that they will develop symptoms of Parkinson's by about 12 to 13 weeks of age. They also have the pathology that comes with it, so they'll have the alpha-synuclein in the brain, they have the inflammation in the brain, so the microglia are getting activated, they have the constipation, and importantly, they have the motor function. So there's some slowness, and they are not quite as dexterous. So it's a mouse, of course, but you can see that there are problems with motor function, with movement. If you breed those same mice that are bound to develop Parkinson's in germ-free, that is entirely sterile conditions, so they're born by C-section and then they're being kept completely germ-free, they do not develop Parkinson's. So somehow these microbes are needed for these mice that are they are destined to develop Parkinson's. They do not develop it if they don't have the microbes on board. If you look at the brain, the changes are also fewer. If you take these previously germ-free mice and you now do a fecal transplant, a poop transplant from other mice to these previously germ-free mice, the mice get Parkinson's again. If you go and treat the mice now with germs, so they're these alpha-synuclein overexpressing mice and they have their germs, but by week 5 or 6, you start giving them hammer loads of antibiotics. This is not something that could be done in humans to that extent because we like our microbiome, so we don't want to kill everybody, but that's what they did. So they completely got rid of all the microbes and the mice again did not get Parkinson's. They also looked at the role of these products that the bacteria makes, the short-chain fatty acids, and even the mice who did not have the bacteria, so the germ-free ones, if you just gave them the short-chain fatty acids, they still developed the changes. So there might be something going on there. And so in summary, what the message was from that series of experiments was you have your typical microbiome, and so your typical microbiota. They produce short-chain fatty acids, which act on the microglia, those are the immune cells, and then the changes that go with Parkinson's develop. If you do not have the microbes, you don't produce short-chain fatty acids, the microglia do not go into action mode, and you do not develop Parkinson's. And now to top it all off, if you take these germ-free mice and you now do a poop transplant from a human, it makes a difference as to whether you take that poop transplant from a healthy donor or from a person living with Parkinson's. Because if you take it from somebody living with Parkinson's, you make the mice, the symptoms they develop will be worse. So there's something in the microbiota of the people living with Parkinson's that seem to make those symptoms worse if they're transplanted into mice. Now, this is all in mice, not in humans. So there's a lot of work to be done to look at what is actually going on in us rather than mice. But, of course, it is truly, it's inspiring to see there's support for that at that level. I won't go through this again because I pretty much explained it, but this is, again, our model that we're talking about. I don't think you can really see it well. So we need to address this step, then a miracle occurs, step 2. That's what we're going to work on now. And to do that, we are doing research to hopefully move from the microbiota there in the left corner to the molecules that then communicate with the brain evolving from there. So we've got a study at the Pacific Parkinson's Research Center at UBC going on where we collect stool samples, but we also do nasal swabs, we do oral swabs to take samples of the microbiome that is living in different parts of the body from both people living with Parkinson's as well as healthy controls. And so I just have to say, everybody who's there, who wants to participate, most of them have, of the people who've been eager to participate have Parkinson's. We have difficulties recruiting controls. So if there's anybody you can think of who is a potential healthy control and who might be interested in that, please let us know, because we really need healthy controls. Without that, we can't interpret, we can't compare to how is this different to Parkinson's. So healthy controls. We're looking at the bacteria, but we're also looking at fungi, which hasn't been done before, so that is a very novel part of that. We're looking not only is there Parkinson's or not Parkinson's, but we're also looking at the different symptoms that come with it, particularly motor symptoms, but also non-motor, such as, of course, constipation, but also things like depression, anxiety, fatigue. We are going to look at what we call metabolomics, which is kind of the chemical fingerprint that you can get from the blood of all the biological processes going on. And there can be, for example, pathways be identified where the microbes might have an influence. And we're looking at other influencing factors, such as diet, of course, that is a very, very important part of that. So you get to fill in a rather detailed diet questionnaire. The whole thing doesn't take more than 2 hours, so don't worry. Looking at things like pets, this is our Oscar here. He's got a lot of microbiome, I tell you. Medications, other medical conditions, demographics, etc. So there's a lot of information that needs to be collected. So what would it mean for people who participate in the study? So you come for one visit so far. There might be, if we get more funding, follow-up visits in future, but for now it's one visit. You do clinical assessment, which is mostly filling in questionnaires, but it's also the usual motor assessment, movement assessment that we do. And then we take the swabs. You can provide the stool sample at home. That's a little kit. Everybody so far, is our feedback, has not had any problems collecting that. It's a tiny little, it's a vial that you send back. You get a little package that is already pre-paid and addressed to us, and you just really need to send it off and mail it back to the lab, and then we'll go and analyze things. So far it's it's off 2 hour or so visit, and that's it, hopefully with some follow-up later on. Who can participate? So of course individuals who have Parkinson's as well as healthy controls. We seek people who've had their onset of Parkinson's in the last 10 years, but this is not a completely hard rule. If somebody is very keen, we might be able to look at that. Age of onset should have been between 40 and 80 years, just to keep the group fairly, homogeneous, or fairly the same. Exclusion criteria, so if there are any gut problems, so if somebody has Crohn's or colitis, or any of those inflammatory gut disorders that would haze the waters, so to speak, we wouldn't quite see what is due to Parkinson's, what might be due to a gut disorder, or if you have the flu that very day, that wouldn't work, so an active infection wouldn't work, or if you're taking steroids or other medications that influence the immune system, that would of course interfere with the results as well. But otherwise, you're very open to comers. Potentially, if this line of research, and this is not just our research, but generally speaking, if we continue to get these encouraging results, we're hoping to understand more about the pathophysiology of Parkinson's disease, possibly also very early on. We hopefully could maybe develop some tests to see if somebody has a specific microbiome composition that might be a higher risk of Parkinson's. And the goal, of course, is always to develop treatments. And treatments in this case could be things like probiotics, which you already can buy in the health food store, but obviously would make them a bit more targeted and make them more sophisticated, as well as interventions with diet. So really, it's in many ways down to the basics, which is really nice because it's stuff you can do yourself, but we really need to get the information to make specific recommendations. And with that, I would like to just introduce you to our team at UBC. We have Mihai and Kristen there on top. Kristen is our research coordinator. She'd be working with you. And we've got Mihai, who's our PhD student working on the project. We work closely together with Dr. Brett Finley, who is the microbiome expert. He's really a world expert in that, very much leading in the field, who's doing the analysis. We've got Dr. Brian McVicker, who is the head of, or co-head of the Center for Brain Health and is a neuroscientist. We've got a very large team of expertise from many other disciplines that play a role here. And again, a big thank you. Without you, we could not do any of the research. And therefore, big thank you to the Parkinson's Society of BC. And with that, I'll thank you for your attention, and I'm happy to take your questions.
I'm going to try and hold the question.